So this is our big scheme, okay, where pretty much everything we have discussed this semester kind of makes sense and how everything gets summarized. So I'm not going to go over it again, but you know what I'm talking about. Now, with regards to human metabolism, which is going to be our focus today, it's important to know that most of our reserves are stored in fat, which is not useful for gluconeogenesis, as we have discussed, right? Because if you break down fat, at least the dogma says that you end up having acetyl-CoA, and well, you do get, if you break down fat, you get acetyl-CoA, but that's a fact, that's beta oxidation. It appears that in most cases, you cannot use the acetyl-CoA to generate glucose. It's not gluconeogenic. We do not have the isocitrate lyase, that version of the glyoxylate SANT to be able to generate carbons that are more than 2 carbons in length. So we cannot get to, let's say, 4 carbons out of 2 carbons in the TCA cycle. There is some new evidence that some cancer cells somehow can do this, but through a different way, which we will discuss. And our other major reserve is, of course, amino acids that are stored in protein, in muscle for the most part. And that, when it's broken down, protein, when it is broken down, you can get a lot of glycolytic intermediates, as we saw in a couple lectures ago. However, so this is where our reserves are, right? Fat and protein, but many tissues really rely on glucose breakdown. You really need to have glucose around. Brain is a classic example. Just brain alone will need about 100 to 150 grams of glucose every day. So it's an extremely glucose demanding organ. On top of that, when we're talking about diet, we have to also consider the fact that we do not make, despite all the complexity of the amino acid reactions, biosynthetic reactions that we discussed, we humans can only make about half of them. So the rest must come through the diet. Okay. As I just said in the previous slide, it is thought that fatty acid carbon, you cannot use it to generate gluconogenic or other higher order compounds. However, it was shown that in endothelial cells, you can get carbon derived from fatty acid breakdown. You can incorporate it into nucleotides. Remember the reactions we talked about last time in nucleotide biosynthesis. So this is happening, especially in some cancer cells, which generate power generation of blood vessels and endothelial cells. They can proliferate. So somehow fatty acids come into the mitochondria, which again, remember that the rate limiting step for fatty acid breakdown, fatty acid oxidation, is actually the carnitine shuttle, which determines how fast you get them into mitochondria. They get burned down to acetyl-CoA, and somehow that carbon from acetyl-CoA can find itself in nucleotides and DNA. Okay, so this, especially in proliferating and endothelial cells. So that seems to run counter to the dogma, as I said, that fatty acids cannot be the source of, let's say, gluconeogenic or other higher carbon compounds. And just to illustrate again that for carnitine phosphate, for fatty acid breakdown, the carnitine shuttle is rate limiting for beta oxidation of fatty acids. So how could that be then? Is it possible to actually generate at least some gluconogenic substrates from fatty acid breakdown in human cells? We know microbes can do it because they can run the glyoxylate shuttle SANT in the TCA cycle, but we don't have these enzymes. Well, it turns out instead we think that what's happening is the canonical structures, the canonical pathways that we've discussed of TCA cycle and shown here for humans and glycolysis. again, for organisms that have the glyoxylate sum, that's possible, but we don't have these extra reactions. Okay, we are more the prototype shown here in A. Well, it turns out if you actually burn fatty acids, you do get acetyl-CoA, but some of this, through the ketone body formation, you can actually make acetoacetate, which we know will give rise to acetone. And acetone, as it turns out, can get out of the mitochondria. And through these reactions, acetyl, so we have seen these reactions. These are really the ketone body formation. But what happens to acetone here, it can get all the way into pyruvate through these reactions, okay? Now that's a model, and once you get to pyruvate, of course, then you can go backwards in glycolysis, and that might explain how it is possible, at least in some situations, to use fatty acid carbon to get to gluconeogenesis. And this actually is the review of gluconeogenesis, the pyruvate. You know, you have to really get to oxaloacetate and phosphenol pyruvate, get it out with the salts we had discussed, and then go all the way back to glucose. So theoretically, at least, you know, this is a theoretical paper. It's in silico evidence, not necessarily actual evidence, experimental wet evidence. But theoretically, it seems that there is a path. There might be a path. Okay. Still, despite all this, it's proper to say that from fat you cannot really get, you cannot satisfy your biosynthetic needs to make glucose or other amino acids or things like this. In terms of reserves, again, I have shown you something similar in another slide. This is how much we have a huge amount of energy reserves stored in fat. If burned down in terms of how much energy you will get from protein, you get a good chunk, nothing close to fat, of course, but it's still very substantial. In contrast, look at glycogen, which we rely on it every day, right? We make and break down glycogen all the time. Very little. But the little there is, it makes a huge difference. So this is fat reserves, protein reserves, and carbohydrate reserves. So for all these considerations, you cannot just eat fat. Human diet must contain glucogenic fuel. Okay? We must give our bodies things that they can use to make glucose. Even the normal overnight fast, when we sleep, some muscle protein is broken down every night. The synthesis of this protein, okay, it's broken down. So how do you make it up? Well, you have to have some protein in your diet. Okay, so because if you just eat, let's say, fat again, you won't be able to resynthesize that protein because 10 amino acids are essential. So you really have to take real protein intake every day. So protein intake is essential for to maintain a living human. If you this is the overnight fasting that we're talking about. Now, if you go to longer, prolonged fasting, days or weeks, then it really, you cannot, okay, you're going to waste a huge amount of your muscle, so you're going to destroy it, but at some point it becomes intolerable. You can't go below a certain threshold. And at that point, even the brain will switch exclusively to start using ketone bodies. So you won't have to destroy all of your muscle. How much can you lose? You can lose pretty much a quarter of your weight if you're an adult with no harm, okay, or very little harm, I should say. So that's not really a big deal for adults. But it's a big problem for children, right? Children cannot fast. Protein intake is a must because they have to make new cells, they have to make new tissue, they have to grow, right? So for that, you really need to have a continuous supply of protein. No prolonged fasting for children. All right, so let's go back to this. The take-home message, protein is important. Okay, we have to have a constant protein component in our diet. So that's the absolutely most critical thing. And fat does not provide really, despite some in silico evidence and some other experiments that have been done in some specific cases, fat alone can only give you ATP, can only give you energy, but no more. Okay. Now, we're going to switch gears a little bit and talk about the concept of the energy charge. What is that defines the energy status of the cell? We have talked about many times how the ratio of ATP and ADP and also NADP and ADP is this energy and electron currency of the cell, how they couple really basic metabolic blocks. And the coupling is shown here. Let's go back to the first slide, sorry. Look at these molecules, okay, they're right there, ATP, NADP, NADH. These are the things that in yellow boxes that really couple these fundamental metabolic blocks, whether it's a catabolism or anabolism and the various aspects of it, right? So those are extremely important molecules. Now, why ATP is an energy currency? Because it's got this high energy phosphate anhydride bonds. This is an old slide from the introduction. What is it? It's really two phosphates bound to the same oxygen. That's an anhydride bond. And these are the high energy phosphates right there, okay? With a, depending if you have one phosphate that you're AMP, you don't really have any high energy bonds, but if you're ADP, you have one, you can break down this bond, and if you have ATP, you have two high energy bonds. So if you define, so this values gives you a value of the energy charge of a single molecule. ATP counts for 1, half for ADP, and 0 for AMP. So if you measure all these things in the cell, you can get some idea of your energy status, and that's the concept behind the energy charge, which is defined as the fraction of the nucleotide pool that carry phosphate anhydride bonds. So it's you have all these nucleotides, ATP, ADP, and AMP, but the question is what fraction actually has the phosphate anhydride bonds? As I said, if you have only ATP, then you have the highest energy charge, because you got, for every molecule of ATP, you have two high energy phosphate anhydride bonds. So you can define that, your energy charge will be the concentration of ATP plus half of the concentration of ADP. So you get a full complement for ADP, half for ADP, and then the denominator will be the sum of all these states, okay, of adenine nucleotides. Of course, notice that, on the numerator, there is no AMP, because again, the energy charge for AMP is 0, right? As we have discussed also in the introduction, ATP hydrolysis is favored. This, if you actually look at the ratio, it's ridiculous, okay? It's 10 to the minus 8, but yet in cells, okay, cells accumulate more ATP than ADP. Okay, so there's at least twice as much as 10 times more of ATP compared to ADP. So the relative pools in living cells, not dead cells, so they go through extremes to maintain these relative abundances. There is a lot more ATP than ADP or AMP. AMP, in fact, is a tiny amount. But what this means is that some changes in ATP alone will be hard to be detected, okay, because you have too much of it. So how does the cell know if its ATP levels are dropping? You need something more sensitive. And it's actually the ratio of ATP to AMP that's important for regulation, because small changes in AMP will have a profound change in this ratio. Okay. So because AMP levels are kept so low. So that's the whole idea why AMP is a sentinel metabolite. It lets the cell take an accurate gauge of its energy status. So in general, as I said, cells keep high energy charge. Okay, so they try to keep it between 0.8 and 0.9. And it's also important to remember the important role of adenylate kinase, an enzyme that takes 2 ADPs to make 1 ATP and 1 AMP. Adenylate kinase does not alter the energy charge. It just returns nucleotide concentrations to equilibrium, okay? And so they properly reflect, so you have a proper representation of these relative pools right away. So a pegylated kinase will do that. All right. And at equilibrium, you want this to be about 1. That's the equilibrium. The ratio of you multiply ATP by AMP. These are the products over the square of ADP concentration, and that would be about 1 in equilibrium because of this equation here, chemical reaction. All right. So if you look at the relative abundance of ATP, ADP, and AMP, okay, as a function of energy charge, shown here on the x-axis, when you have a very high energy charge, and remember, this is where we are for the most part for most cells, between 0.8 and 1. This would be the abundance of the relative abundance of ADP and AMP. So AMP would be very low, going from even from 0.7 to 0.8. You have a twofold drop at AMP levels. So that is why AMP is such a good sentinel metabolite, right? All right, so enough with energy charge. We defined it. What does that mean about regulation of metabolic pathways? Well, it turns out, okay, that the higher the energy charge, so the more you are on this side, on the right side of the x-axis, the more you activate anabolic reactions. Does that make sense? Of course. I mean, this is what you need for anabolism. You need ATP and reducing power, and ADPase. So the more ATP you have, the more it makes sense to activate anabolic reactions. Conversely, the higher the energy charge, the more you suppress, catabolic reactions. Okay, so you don't generate more. In contrast, if your energy charge is low, this is when catabolic reactions are activated. Does that make sense? Of course. If your energy charge is low, the signal tells the cell to activate all these catabolic pathways, let's say glycolysis, the TCA cyclone oxidative phosphorylation, to make more ATP, right? So that's a general principle of regulations. In general, that's how the reactions will be regulated. So all the catabolic pathways in general will be activated by AMP. Why? Because when AMP is high, it means the energy charge is low. Sometimes they are repressed by acetyl-CoA or phosphoenol pyruvate. These are intermediates, of course, as we know in these pathways. So AMP will activate catabolism, will activate breakdown, okay? Anabolic pathways, on the other hand, as we saw, we have many examples of this in when we're talking about biosynthesis pathways, let's say of amino acids, etc. Usually what you see are the specific end product inhibition. If you make too much tryptophan, you're going to go and inhibit the very first enzyme of tryptophan biosynthesis, and so on and so forth. So you get these things happening. So whatever products you are making from anabolins will go on and inhibit the specific pathways that made this product.